Panax notoginseng is a highly valued perennial medicinal herb mainly grown in Yunnan, China. In 2022, a new leaf spot disease caused by the fungal pathogen Borrelia azigua was observed, affecting about 20% of plants in Quebec country. Symptoms include dark brown leaf spots, which can cause and compromise plant health. Borrelia azigua has a broad host range impacting more than 200 plant genera worldwide. Understanding the genome of this pathogen is crucial to uncover this pathogenic mechanism and develop effective disease management strategies. In this study, we report the first complete genome sequence of strain HEIC and investigate key virulence factors, cell wall degrading enzyme, and secondary metabolite gene cluster.
I will explain the rationale of this study. Leaf spot disease is an important issue because it affects Panax notoginseng, which is an valuable medicinal crop. When a disease affects a plant like this, it can reduce both quality and safety. So it becomes important to understand what is the causing this infection. Another reason this study matters is that Borrelia azigua was still poorly understood at the genomic level, so there was a clear gap in knowledge about how this pathogen works. Moreover, genome information is useful because it can show virulence-related features such as C enzyme, effectors, and other genes linked to the infection, which can help to explain how the fungus infect the host and guide future disease control. The main objective were to sequence and assemble the complete genome of the strain HEIC, which is the strain causing the disease. After that, the genome needed to be annotated so its main features such as gene, repeats, and non-coding RNAs could be identified. Another objective was to find C enzyme involved in plant cell wall degradation because this enzyme may help the pathogen break into the plant tissue. Finally, the study aimed to predict virulence-related elements such as effectors, PHI gene, and secondary metabolite clusters, since these features are closely linked to the pathogenicity and host interaction. Overall, this objective helped to build the genomic foundation for understanding how B. azigua causes leaf spot disease and how it may be managed more effectively in the future.
Next, I will explain the methodology used in this study to identify and analyze the fungal pathogen-causing disease in Panax notoginseng leaves. First, the researchers carried out sample collection and fungal isolation. Diseased Panax notoginseng leaves were collected from Wenshan, China, where symptoms of infection were observed. The pathogen was then isolated from the infected tissues and cultured on PDA medium, which is commonly used for fungal growth. This step was important to obtain a pure fungal culture for further experiments. Second, a pathogenicity test was conducted to confirm whether the isolated fungus was truly the cause of the disease. Healthy leaves were inoculated with fungal mycelium and incubated for 7 days. After incubation, disease symptoms developed, showing that the fungus had disease-causing ability. This step follows Koch postulates, which are used to confirm pathogenicity. Third, the researchers perform morphological and molecular identification. Morphologically, they observe colony appearance and microscopic fungal structures, such as spores or and hyphae. For molecular identification, ITS and RPV2 gene sequencing were carried out. These gene regions are commonly used in fungal taxonomy because they provide accurate species level identification. Next, the study continued with DNA extraction and whole genome sequencing. Genomic DNA was extracted from fungal mycelia. The researchers then used both Illumina and Nanopore sequencing platforms. Illumina provides highly accurate short reads, while Nanopore produces longer reads that help improve genome assembly quality. After sequencing, the researchers carried out genome assembly and annotation. Low-quality reads were filtered out before assembling the genome. Functional annotation was then performed using databases such as KEGG, GOKOG, and SwissProt. These databases help predict the functions of genes and identify metabolic pathways and cellular processes in the fungus. Finally, the researchers performed pathogenicity-related gene analysis. KZ analysis was used to identify carbohydrate active enzymes, especially enzymes involved in degrading plant cells walls, which help the fungus infect host tissues. In addition, PHI-based and anti-smash analysis were used to identify virulence genes and secondary metabolite by synthetic clusters. These analyses provided insights into how the pathogen causes disease and survives in the host plant.
The fungus causes dark brown spots on the leaf of Panax notoginseng as is shown in figure 1a. When grown in the lab, its color changes from green to dark brown over 30 days as is shown in figure 2a until F. Under the microscope, you can see brown spores and septate hyphae as is shown in figure 2G until J. Finally, DNA tests confirm that this fungus is Bioremia exigua as is shown in figure 3. The genome of Bioremia exigua is 26.35 MB in size with a CG content of 53% and it is assembled into 25 contigs. Repeat sequences make up about 5% of the genome. A total of 11,701 genes were predicted and 98% of them have known functions. Additionally, the fungus possesses 412 CAZY genes which help it breaks down the plant cell wall. The most common types are glycoside hydrolysase 159 and auxiliary activity enzyme 142 as shown in figure 8. The fungus has 252 secondary metabolite gene clusters such as NRPS and PKS shows in figure 9. It also contains 3,323 pathogen host interaction genes including 47 effectors, 91 virulence enhancing gene and 98 lethal genes as seen in figure 10. Using DNA and genome comparison to the correctly identified Bioremia exigua, formerly Forma exigua, this study found that its genome is about 26 MB with 11,701 genes, that about 800 is repeating DNA, which helps adaptation but may complicate disease control, that it has 412 cell wall degrading enzyme plus 47 effectors and 91 virulence genes, that 8 infection, and that this first complete genome provide a foundation for understanding and managing leaf spot disease.
The complete genome of Bioremia exigua strain HEIC was determined to be approximately 26.35 megabases containing over 11,000 predicted genes. 412 putative CAZY genes were identified, indicating its ability to degrade plant cell wall. The genome also contained 252 secondary metabolite gene cluster and 47 predicted affected proteins, revealing complex pathogenic mechanism. PHI-based analysis further highlighted genes associated with virulence and lethal phenotypes. Overall, these genomic insights provide a solid foundation for understanding pathogen-host interaction and developing targeted strategies to manage leaf spot disease in Panax notogenese.